Welcome everybody to this webinar. My name is María del Mar Saez, and I'm a product manager at Cytognos. Today I'm very happy to present our invited speakers, Eduardo López Granados and Luz Yadira Bravo Gallego. They are going to talk about the capabilities of the flow cytometric OX40 assay to determine specific SARS-CoV-2 T cell responses. Eduardo López Granados is a medical doctor and specialist in immunology, with a PhD in biochemistry. Currently, he is the head of the Clinical Immunology Department at La Paz University Hospital in Madrid, Spain. He held positions as postdoctoral research fellow at the National Institute of Allergy and Infectious Diseases and also as senior clinical lecturer in clinical immunology at the University of Oxford. Dr. López Granados is the director of the Lymphocyte Pathophysiology Group at La Paz Biomedical Research Institute and of the Rare Diseases of the Immune System Group at the Spanish Rare Disease Network Research Center. He is the coordinator of the Network Research Working Group of the European Reference Network for Pediatric Transplantation, and since 2011 he is also a member of the PID group of the Euroflow Consortium. He is deeply involved in the development and clinical implementation of novel diagnostic tools for the analysis of cellular and antibody responses to SARS-CoV-2 infection in healthy and immunocompromised patients. Luz Yadira Bravo Gallego is a medical doctor and specialist in pediatrics, master in pediatric infectious diseases, and specialist in immunology. Currently, she is the medical advisor of the European Reference Network for Pediatric Transplantation, and she is part of the Lymphocyte Pathophysiology Group at La Paz Biomedical Research Institute and the Rare Diseases of the Immune System Group at the Spanish Rare Disease Network Research Center. She is an expert in the clinical and immunological evaluation of the host-pathogen interaction in several clinical scenarios, with particular dedication to the analysis of the immune competence in pediatric patients that receive hematopoietic and stem cell transplantation, in which she is focusing her PhD project. We are so excited to have you both here today, Eduardo and Yadira, so thank you very much and welcome.
Hello everyone, thank you for attending this webinar. My name is Eduardo López Granados and I would like to thank Cytognos for organizing this webinar and inviting us to speak about our experience with the use of the OX40 assay for the flow cytometric determination of specific SARS-CoV-2 T cell responses. I will give you an introduction about our experience with the use of antigen recall assays and the particular utilities or capabilities of OX40 assay. And then my colleague, Yadira Bravo, will go more into detail with our more direct experience in the application of this test to the analysis of patients that suffer the SARS-CoV-2 infections and our first results from the pilot study in vaccinated individuals. So you know that there are many options to analyze specific antigens, either whole proteins or lysates or peptide specific antigen responses in the laboratory. Obviously, each of the different methods have their own advantages and disadvantages. You know that usually the rationale behind this is that once we have generated in a first exposure specific memory T cells, we can re-stimulate these cells with the antigen for which they are specific, and then we can use different readouts to explore the presence and the different type of effector responses in this recall activation to different antigens. You know that probably one of the most used readouts is the production of interferon gamma that can be detected either in supernatants, like for instance in the cytokine secretion assays, in which we can stimulate cells in culture, collect supernatants and analyze them either by ELISA or more recently by different methods, even some multiplex methods that allows the analysis not only of interferon but other cytokines simultaneously. We have another methods that are quite extended for the analysis of cytokine production of these cells. There is the intracellular cytokine staining by flow cytometry, which also, especially with the latest flow cytometries that allow the simultaneous analysis of multiple parameters, could be very comprehensive in analyzing the different types of effector responses attending to the production of cytokines. And then a very robust and very standardized method that even has been developed for the diagnosis of, for instance, Mycobacterium tuberculosis infection is the so-called ELISpot assay, in which we detect the cytokine production immediately just in the vicinity of cells that have been attached to the bottom of a plate. And this is a quite robust assay, but that so far, except for some recent developments, usually allows the detection of only one parameter, as it is interferon gamma production. Obviously, we have the more specific capacity of identifying circulatory specific T cells, which will be the use of different structures that just mimic the class 1 and class 2 major histocompatibility complex, and then with different specific peptides, we can directly stain specific cells, just attending or just mimicking the recognition of the HLA molecule and the TCR. This would be the more specific method, just to stain, as with any other region, specific T cells. So this tries to be a comparison of the different methods. I didn't mention before the most, let's say, traditional, or the initial assays to measure responses, specific antigen responses, which is the in vitro proliferation, either with deuterium or with, more recently, with some markers that allow to trace the different divisions of the cells. Advantages of this assay is that, well, it can be done, is highly sensitive. We can detect small percentages of cells specific for the antigen. It takes some time to get the results. We need some proliferation in the culture, so we have to wait at least 5-7 days to have results. We can use different type of antigens like lysates or even peptides, although usually lysates is the most used antigen source. And then we are going to measure proliferation. What's the disadvantage? That because we have all this step of proliferation, probably is less quantitative, and we can only infer the amount of cells in the population of origin, because we have several cycles of proliferation. I mentioned the cytokine release assays that we can use, either measuring responses in the supernatant by ELISA or ELISpot. Here we can use whole blood for some methods that are based on the detection of cytokines in the supernatant of the cultures, or ELISpot, in which we will have to purify PBMCs. We have different times of incubations, from 6 hours minimum to 24 hours, depending on the conditions. We can use whole antigens or whole proteins, but usually we can employ different peptides, either cocktails of peptides or specific peptides. If we have some idea about the immunodominant peptides and we have the HLA type of the individual, it is known. But usually when we use peptides, it's more just to without knowing or without specifying initially the HLA type, so we use cocktail of peptides. And as I said before, there is more versatility or more flexibility with the ELISA's methods, in which we can use even multiplex systems to analyze different cytokines, whereas in ELISpot so far it's mostly interferon gamma which we can measure, although recently there are some fluorescence-based developments in which we can simultaneously measure more than one parameter. And there are highly sensitive methods, and even has been validated, like the quantiferon, for instance, assay for the measure of responses to Mycobacterium, or also ELISpot for the measure as well of Mycobacterium tuberculosis infection. What are the disadvantages? That we cannot identify a specific CD4, CD8 responses unless we use peptide specific for one or the other, but usually there is not a possibility to discriminate the cytokine production if it corresponds to CD4 or CD8 cells. In contrast, we have the intracellular cytokine staining assays, which are based on flow cytometry. Probably these are the most extended in the research settings. We have to start with PBNCs in order to increase the quality of the staining. We have as well an incubation time from 6 to 12 hours. We can use combination of peptides, and the interesting addition to the previous methods is that here we have the opportunity to measure different and a high variety of effector phenotypes by the production of the types of cytokine productions. There is another version that is the one that we will just discuss today, which is the use of activation-induced markers in the surface of the cells. In this occasion, we can start also with whole blood or PBNCs. We have a slightly longer incubation time depending on the marker that we want to analyze. Some are markers that have a peak of expression in 12-24 hours, and so others we will have to wait up to 48 hours. Sorry, this is a mistake. This should be 48 as the one where we are going to tell us about it today. That is the OX40 assay. And as I said, there is a potential to combine both methods in which we can use PBNCs to stain simultaneously for surface markers and intracellular cytokines, and then this would be a very powerful capacity to analyze different effector and different cell types. So what's the disadvantage of, for instance, intracellular staining? That is quite labor intensive. The protocol is much longer, is more sensitive, we have to look more carefully for good combination of antibody to detect intracellular cytokines, and probably is not very suitable for very frequent use, or if we are dealing with a large number of samples. And then, as I said before, the use of the tetramer staining technology or any other multimers to detect by flow cytometry the specific T cells requires the use of PBNCs. We can have some results in a very short time, just like any other flow cytometry staining in surface. The disadvantage is that we need to know the HLA type of the individual in order to load the tetramers with different specific or usually monodominant peptides that are more or less known, and then this is very a powerful approach to detect without any stimulation of any proliferation cycle specific T cells, but it has the difficulties of the need to know the HLA type and have a large array or a large collection of different molecules of different tetramers in order to being able to address any specific haplotype in the patients. As it was mentioned before, obviously, as being just surface staining has the potential to combine these with many other markers. We have the opportunity to really identify quite nicely different characteristics of the cells. So keeping this in mind, now we will explain about the OX40 molecule. So OX40 is a T-cell co-stimulatory molecule that belongs to the family of the TNF cytokines, the cytokines family. It is expressed in T cells upon TCR stimulation, and it has a peak of expression of 48 hours in vitro or 3-4-5 days in vivo. Then the kinetics of expression of OX40 is a little bit longer than, for instance, other activation molecules of T cells like CD69 or CD40 ligand. And OX40 ligand, which is the ligand for OX40, is expressed in professional APCs. We know that it's important, these molecules, this OX40 ligand interaction is important for the generation of memory T cells, and this has been addressed in knockout mice studies, in which we see that the lack of these proteins lead to a very severe affection of memory T cells, and even more recently in the description of patients with defects in these molecules. So the interaction of OX40 and OX40 ligand provides a signal for the T cell expansion that is crucial for survival of effector T cells. And the interesting thing is that these molecules, as I said, is upregulated in memory T cells, in contrast, for instance, to CD28, which is expressed constitutively in all naive T cells. Therefore, the upregulation of OX40 is more specific for the identification of memory T cells. So the OX40 assay for detection of antigen-specific CD4 cells is a simple technique that consists in culturing cells stimulated either with antigen or mitogen for 48 hours. If you remember, that's the 48 hours is the peak of expression of OX40 in the membrane of the T cells, and then combined with the detection of the regulation of another activation marker, which is CD25, the receptor of interleukin-2. And then the combination of both allows the identification of antigen-specific CD4 cells, which does not limit the decision, the prior decision on what combination of cytokines will be needed to detect memory T cells. This prevents the bias of the detection of only some types of effector cells, and also the combination of CD25, CD34 is a good combination to rule out the possibility of detecting regulatory T cells which express CD25, activated regulatory T cells that can express CD25 but do not express CD30 or CD34. So usually in any individual, it has been estimated, and some results from the original descriptions of these assays more or less show that there is a small proportion of CD4 cells in the peripheral blood of any control individual co-expressing to CD25 and CD34. So there is a very slow, very low background, and then is a good assay really to detect recall antigen responses. And then this has really to be a sensitive and a specific assay, and it has been applied and has been used in different studies to, for instance, analyze the responses to different bacteria and viruses, like, for instance, cytomegalovirus, hepatitis C, HIV, and Mycobacterium tuberculosis. And even there are some studies using this assay to analyze responses to vaccinations, for instance. So the original description of this assay made by the group of Sanders, they just demonstrated how there is a good correlation of this co-expression of CD25, CD34 with there is a capacity to detect different antigens specific responses. As I mentioned before, this is just one of the original slides from the first paper, in which they show a good capacity of detect and specifically activated T cells with PHA, with mitogens, or with different even some superantigens. We have experience using this assay with anti-CD3 specific TCR stimulation as a control, as a positive control. And here you see how it has been applied to the detection of different antigen-specific responses, as I said before, to tetanus toxoid, flu, candida, cytomegalovirus. This is just a collection of different results from different healthy individuals to demonstrate that the optimal time for the detection of the OX40 expression in the surface of the cells is at 48 hours. It can still raise in the next hours, but it's important to do it at this time as well, because one can see proliferation usually upon 72 hours in culture. So analyzing cells upon activation, for 48 hours upon activation, also will preclude the analysis of proliferating cells. And then there is a good correlation with this assay, the OX40 assay, with other antigen-specific recall assays, like, for instance, the lymphoproliferative assay that was used originally as a gold standard. And you can see here that without the stimulus, there is no proliferation of the same cells when compared the proliferation in response to in resting conditions, and for instance, in response to a cytomegalovirus specific antigens in a cytomegalovirus negative individual. But for instance, you see that there is a good correlation of the proliferation with tetanus toxoid and Mycobacterium tuberculosis peptides when comparing the proliferation with the upregulation of CD25 and OX40 here in these quadrants. Also, it was important to correlate the expression of the co-expression of OX40 and CD25 with the production of cytokines, and these results as well from the very original description of this, the proposal of this assay to measure antigen-specific responses. It was also analyzed within the population of double positive CD25, CD48 activated CD4 cells, how a good proportion of these cells were expressing interferon gamma, whereas in the single positive CD25 or OX40, CD34 single positive cells, the production of interferon gamma was very low. And on the contrary, if we activated cells and we gate on interferon gamma producing cells, like for instance in CD4 cells, the production of interferon gamma upon activation, you see how a huge proportion of the interferon gamma producing cells are co-staining for CD25 and CD34. Meaning that there is a good correlation between the upregulation of these two co-stimulatory molecules and the production of cytokines. And it was also important to show that using some transwell culture studies that the upregulation of these molecules do not seem to be due to a bystander effect. There was some studies, some experiments just using transwell cultures in which two different populations of cells were activated, one of them with the specific peptides, and it was mentioned in the other side of the transwell that just through the sharing the supernatant, there was not a specific activation in the cells that have not been directly exposed to the antigen, meaning that the effect of bystander activation effect is minimum. So why do we think that it's important to develop, or at least to have the opportunity to use antigen-specific T-cell responses in the current SARS-CoV-2 pandemic? It may not be easy to do it in a large scale, but there have been several attempts to detect by serology the prevalence of infected individuals in the population, but we know that only measuring antibodies may not be sufficient to estimate prevalence. There may be some individuals that do not produce antibodies in response to the viral infection, and that has been shown in different publications, but also the antibodies can decay, the levels of antibody can decay in the peripheral blood, and the inclusion of together not only measuring antibodies but also to detect specific T-cell responses could be the best way to estimate the number of individuals exposed to the virus and the infection. And then we are a center that we are specialized in the diagnosis and therapeutic approach and treatment of patients with primary and secondary immunodeficiencies, and also we have to estimate the status of the immunocompromised status of different individuals in our hospital that are either subjected to transplantation, or they are under immunosuppression treatment for autoimmunity, or due to oncological processes. And then for us it's important not only infected individuals with vulnerable individuals with one of these conditions, but also when eventually these patients will be hopefully vaccinated, we will have the need to somehow estimate what's the immunization status of these individuals. Obviously these assays are not designed or are not developed for diagnosis of infection, but in our setting, at least to identify groups of individuals that could be a high risk of not really generating a good immune response and not being immunized upon infection or after vaccination, it would be important to at least have the opportunity to estimate this status in order to reassure them when they have suffered the infection, if they will be vulnerable for reinfections, or even to know if the current schemas for vaccination will be useful in immunosuppressed individuals. There's still a lot of uncertainties in this area, and not for massive application, but we are just developing these techniques in order to have an array of techniques that could allow us to explore a bit further some of these circumstances. And then we started in our department with exploring different opportunities. This is just some bits of our experience with the use of intracellular staining for the detection of polyfunctional T-cell responses to SARS-CoV-2 peptides. And this is to give you just an overview of how comprehensive these assays could be. This is just some results from an individual that suffered the natural infection of SARS-CoV-2 in a moderate with a moderate clinical impact. And then you can see here how using a peptide pool of overlapping pools of the nucleoprotein, membrane, and spike protein of the virus, we could detect. This is just resting conditions without any activation. This is just a specific stimulation with anti-CD3 anti-CD28 co-stimulation, and you can see how we can detect cells that produce either interferon gamma, interleukin 2 in response to different peptide cocktails. Obviously, the more parameters we analyze, the more variety we can detect, and it is known that the competency to respond to a virus depends on the acquisition of what is called the polyfunctional cells that are able to simultaneously exert different effector functions. And for instance, we explore, and we are about to publish this data in a cohort of patients naturally infected. We decided, for instance, in this assay to combine the intracellular staining with interferon gamma interleukin 2 with the activation markers like CD40 ligand and CD17A for the detection of the granulated cells. So you can see here in one individual how wide is the spectrum of responses we can detect just attending to subpopulation of T cells that were either able to just exert one of the functions, what we call monofunctional responses, or the polyfunctional responses. So these tests are quite comprehensive, but also, as I mentioned in the introduction, they are very labor intensive, and also sometimes it's difficult to really conclude exactly what would be the best way to, or what would be the population that somehow one's expect to respond homogeneously in all these individuals. So then we decided to explore, as I said before, another assay that could be more less labor intensive, that could not give us some in which we didn't have to decide initially what would be the best readout for the upon activation of the cell. So then is when we move and we explore the possibility of using activation-induced markers. We had experience in the use of OX40 for the responses in immunocompromised individuals to EBV, to Epstein-Barr virus, and then we adapt our previous experience with the new with this new viral infection. So it's difficult to decide exactly what will be the first or what will be the best assay to use. And as I said before, we thought that the activation-induced assays, the markers assays, could be quite powerful to detect rare antigen-specific CD4 cells, and it will combine sensitivity and also robustness in the performance, and also it will be something that we could accomplish in a routine setting, like is our clinical immunology department. Obviously, in order to use these assays, you need experienced flow cytometers and departments that are quite familiar with the flow cytometry. And this just will be to finish and then to give the word to my colleague, Dr. Yadira Bravo, who will give you more specific details about our particular experience with the setting up of this assay. Thank you very much.
Thank you, Eduardo, for your excellent introduction. Now, the second part of our presentation is about OX40 assay for the identification of CD4 positive T cell specific responses to SARS-CoV-2 peptides. And as Eduardo had already mentioned it, our group was working in ICS approach with the pros and cons, and we started working in OX40 as an alternative approach to CD4 T cell response to SARS-CoV-2 through Sanders protocol. Briefly, whole blood were collected in sodium heparin anticoagulated tubes, and PBMCs were isolated by the fecal density gradient method and used immediately. Cells were aliquoted into walls of a 96 wall plate at a total 1 million cells per wall. For each assay, 5 conditions were used. First, no exogenous stimulation or an stimulator or negative control. Second, human anti-CD3 28-2 T cell activator or positive control. And 3 antigen stimulations with peptide pools, peptidator SARS-CoV-2 Prot M, N, and S. Cultures were incubated at 37 degrees for 44 to 48 hours in a humidified atmosphere of 5% CO2. Following a stimulation period, the cells were stained for 20 minutes at room temperature according to the panel. Active4 kit that contains anti-CD3, CD4 and CD25 and anti-CD134 or OX40 antibodies, and 2 additional markers to characterize CD4 memory, anti-CD27 and anti-CD45 RA or anti-CD45 RO. At least 100,000 of forward cyclate of lymphocytes were acquired in FACSCAN2 or DXFlex flow cytometers, and data were analyzed with FlowJoule or Infinicide. The gating strategy to identify memory CD4 T cells was the next. First, single isolation, and then lymphocyte enrichment, forward versus side. After that, CD3 selection, and finally, CD4 selection. For memory CD4 cells, anti-CD45 row versus anti-CD27 were used. Naive, effector memory, and central memory CD4 T cells were gathered by quadrants. Finally, expression of both CD25 and CD134 or OX40 for a total CD4 central memory and CD4 effector memory. And now, how we did the calculations. Fold increase was calculated as the frequency of expression of CD25 and CD134 cells in the antigen stimulation divided by the frequency in the unstimulated condition. A significant fold increase will be greater than 3. For example, this patient who had COVID-19 2.5 months before of the OX40 with mild symptoms, has a significant fall increase in all conditions analyzed. Henceforth, you can see in the top the 5 conditions culture: unstimulated, positive control, and 3 antigens, M, N, and S peptide pools. In the left side, you can see which type of cells are we analyzing the CD25 and OX40 expression. At the top, total CD4, in the middle, effector memory CD4, and in the bottom, central memory CD4. On the x-axis, we can see CD25 expression, and on the y-axis, CD134 expression. Negative expression is determined according to unstimulated condition, or our negative. So, double positive expression will be on the top right quadrant here, and of each dot plot. It's remarkable to note the number at the top right of each dot plot. It's the fall increase in green, all fall increase greater than 3, and in pink, all those under 3. In this case, our patient had a fall increase of 9.3 in the central memory CD4 against pool S peptides of SARS-CoV-2. We proposed a collaboration effort with Cytometry, and they asked us to perform a first phase of protocols comparison that involved previous explained PBMC's isolation and whole blood sample incubated with independent CO2 culture media. The third condition was for perform a mini bulk lysis previous staining step. Samples from 6 patients with or without a history of COVID-19 were analyzed in parallel. As you can see in the color map, green cells represented that fall increase greater than 3, and pink cells under 3. If the protocol wasn't performed, the color cell was yellow. In general terms, subjects without history of COVID-19 were negatives in both protocols, except for this one. And subjects with a history of COVID-19 had a fall increase greater than 3, but whole blood protocols tended to be higher than PBMC's protocol. Only one patient with history of COVID-19 was negative in 3 protocols, and we will talk about him later. Now, let's see in detail patient number 4. These dot plots targeting on CD4 T cells. In the top, 5 conditions culture, and in the left side, you can see in which protocol are we observing CD25 and OX40 expression. At the top, PBMCs, in the middle, whole block mini bulk lysis, and in the bottom, whole blood. On the x-axis, we can see CD25 expression, and on the y-axis, OX40 expression. Fall increase were similar in PBMCs and whole blood, as you can see here and here, and higher in bulk lysis. These dot plots targeting on CD4 effector memory cells. Fall increase were similar in PBMCs and whole blood, and higher in bulk lysis as in total CD4. Now, finally, dot plots are rating on CD4 central memory. Fall increase were similar in PBMCs and bulk lysis and different to whole blood. And what did we conclude? Each protocol has pros and cons, and in whole blood protocols, the time spent on day 0 is less, but it requires more peptides because the final volume is 5 times greater than PBMCs. In second place, bulk lysis protocol isn't cleaner nor more sensitive than whole blood alone. Furthermore, it's difficult to analyze it and requires more time on day 2. So, after compare all factors, we opted for PBMC's protocol. Now, I'm showing you some practical examples of the samples analyzed so far. Are there differences between the expression of CD25 OX40 in subjects with or without a history of COVID-19? So let's see in the detail 2 subjects. One with history of COVID-19 at the top of the left side in green, and another one without history of COVID-19 at the bottom of the left side in pink. These dot plots are rating on CD40 cells. In the top, the 5 conditions culture. On the x-axis, we can see CD25 expression, and the y-axis, OX40 expression. And both subjects had a significant fall increase here in our positive control under human T-cell activator. But only subject with history of COVID-19 had a significant fall increase under SARS-CoV-2 and pool of peptides. Now, dot plots targeting on CD4 effector memory cells. Both subjects had a significant fall increase under human T-cell activator, but only subject with history of COVID-19 had a significant fall increase under N and S pools of peptides. But what happened with CD4 central memory cells? Both subjects, again, had a significant fall increase under human T-cell activator, but only subject with history of COVID-19 had a significant fall increase under M, N, and S SARS-CoV-2 pools of peptides. Are that differences consistent between COVID-19 positive and negative individuals? So far, we had analyzed 23 subjects, 10 without a history of COVID-19. For example, their IgG against SARS-CoV-2 were negative without symptoms, and when was indicated, PCR was negative. And we have 13 COVID-19 positive subjects. Let's see in detail how was the expression of CD4 CD25 and OX40. Patients with history of COVID-19 are represented in a red circle, and negative patients in blue square. On the x-axis are SARS-CoV-2 peptides, M, N, and S, and on the y-axis, the fall increase. The dotted line corresponds to the fall increase 3. And as you can see, there are significant differences in CD25 and CD134 expression between subjects with and without a history of COVID-19. All negative in blue, all negative subjects had a fall increase less than 3. Only 2 patients with a history of SARS-CoV-2 infection showed no response to the SARS-CoV-2 peptides, in total nor memory CD4. These patients are marking with a green arrow or within a green circle, and we are talking about them later. Now, the expression of CD25 and OX40 in CD effector memory cells. Please remember, patients with history of COVID-19 are represented in red and negative patients in blue. Again, you can see significant differences in CD25 and OX40 expression between subjects with and without history of COVID-19. All negative subjects had a fall increase less than 3, and only 2 patients with history of COVID-19 showed no response to any SARS-CoV-2 peptide. The expression of CD25 and OX40 in central memory cells on the x-axis are the SARS-CoV-2 peptides, M, N, and S, and on the y-axis, the fall increase. The dotted line corresponds to the fall increase 3. Once again, you can see significant differences in CD25 and OX40 expression between subjects with and without history of COVID-19, and all negative subjects in blue had a fall increase less than 3, and only 2 patients with history of COVID-19 showed no response to any SARS-CoV peptide. In this slide, you can see a summary of the responses against SARS-CoV-2 peptides in all 13 subjects with history of COVID-19, and the time between infection and COVID infection and OX40 assay in months in the right side of the table. In the color map, cases with a fall increase greater than 3 are colored in green, and cases under 3 in pink. 8 patients have responded against all 3 peptide pools, and 2 patients showed a response against 2 peptides, like a 12 patient, and only one had one response. Only one had response against 1 peptide, patient number 13. Considering this, it seems relevant to stimulate the samples separately with the 3 pool of peptides, and 2 patients with history of COVID-19 were negative. What about this patient? Patients 3 and 9 were previously healthy. The OX40 assay was carried out month 3 and 6 after COVID-19 infection, respectively. They had mild symptoms and positive PCR. However, in both cases, the IgG against SARS-CoV-2 was always negative. After had been vaccinated, the OX40 assay will be performed after 3 to 4 weeks after the second dose. And we are pending to achieve this time. It's remarkable to note the similarity between the OX40 and the ICS responses. Thank you, Elena, for your data and graphs. And as we can see, the OX40 negative patient here in pink had a monofunctional, in blue, response in ICS. Otherwise, patient 2 had a predominantly polyfunctional CD4 response in the ICS, again, except against NPUL. Intriguingly, in the OX40 assay against N peptides, this patient only showed a significant fold increase in the CD4 effector memory against NPUL. So, another thing, how is the kinetics of CD25 OX40 expression in a patient with history of COVID-19? For answer this question, we perform OX40 assay twice in patient 6. First, for 5 months after SARS-CoV-2 infection, and then 9 months after infection. On CD40 cells, CD25 OX40 expression were similar, except for the response to N peptides. And on CD4 effector memory cells at month 5, the fold increase was greater than 3 against M, N, and S peptides. But the expression of CD25 and OX40 calculated by fold increase was negative against all peptides at month 9 after infection. Finally, on CD4 central memory cells at month 5, the fold increase was significant only against SARS-CoV-2 peptide and near to 3 against M and N peptides. And interestingly, at month 9 SARS-CoV-2 infection, the fold increase was significant against M, N, and S pools of peptides. Now, what happened in a primary immunodeficiency setting? It will depend on the type of defect. But here you can see an example of a patient with a common variable immunodeficiency who had a SARS-CoV-2 infection diagnosed by compatible symptoms and positive PCR. 4 months before perform OX40 assay. Our patient had a significant fold increase against M, N, and S peptide pools on CD4 effector memory cells, as you can see in the middle row. finally, OX40 has something to say about vaccination setting. But yes, our very preliminary data on vaccination setting, we analyzed the expression of CD25 OX40 on total CD4 T cells, effector memory, and central memory CD4 T cells against peptides pool. Patients with history of COVID-19 are in red, and patients without history of SARS-CoV-2 infection in blue. On the x-axis are 2 points of follow-up, pre-vac, previous the first dose, and plus 21 days after the first dose of vaccination, and obviously previous to the second dose. The dotted line corresponds to fall increase of 3. In negative subjects in blue, you can see an upward trend of the fall increase here in CD4 cells and effector memory CD4 T cells between the 2 points. And only 50% of subjects with a fall increase greater than 3, here and here. We only had the opportunity of analyzing 2 subjects with history of COVID-19, but that their fall increase against S pool were not significant in both points. One of them was the patient who had only response to M peptide. Our data are so preliminary, and we will continue working in this setting. And now here we can see an example of memory kinetics in a negative subject, pre and post first dose SARS-CoV-2 vaccine. In pre-vaccine point, here in the left, all fall increase were under 3 in pink, and 20 days after first dose, here in the right, we can see a significant fall increase against S peptides from in effector memory and effector CD4 T cells in the right side of the slide, in the row 2 and 4. Now, we conclude that current SARS-CoV-2 pandemic is imposing a phenomenal challenge to all clinical immunology departments. Many uncertainties about the infections remain even with the unprecedented efforts made so far in basic and clinical research. The course of the long-lasting immunization after natural infection and different types of vaccines is not fully understood in healthy immunocompetent individuals. And there is a need for novel assays and surrogate markers that could help us to understand better the immune status of least vulnerable individuals. Thank you very much for your attention, and of course, thank you to our patients and families, healthy donors, and all immunology team in La Paz University Hospital, COVID-19 outpatient clinics, and to Cytognos for the webinar invitation. Now, so we will be happy to hear your comments and questions.